Hello. Welcome to this Nature.com custom webcast titled Shedding Light on Conformational Changes During Amyloidogenesis. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Redshift-Bio. We'll begin the webcast with an introductory presentation from Dr. Libo Wang, Principal Application Scientist, Redshift-Bio. This will be followed by a full-length presentation from Dr. Valerie Daggett, Professor, Bioengineering, Biochemistry, Biomedical and Health Informatics, Chemical Engineering, Molecular Engineering, Neuroscience and Biological Physics, Structure and Design at the University of Washington, USA. We'll then move on to a question and answer session with the speakers. To ask a question, just type it in where it says type your question here, and then press submit at any point during the webcast, and we will answer them today. And now, over to Libo.
Hello everybody, welcome to the webinar. We are very glad to have Dr. Valerie Daggett as our speaker to talk about the conformation changes of amyloid peptide during amyloidogenesis. In her story, a novel infrared spectroscopy method, Microfluidic modulation spectroscopy, in brief, MMS, was used to analyze the structural characteristics of amyloid beta peptides during the aggregation process. MMS was able to detect the non-standard alpha sheet structure, which strongly support Dr. Daggett's alpha sheet hypothesis towards understanding the toxicity of amyloid peptide aggregates. Before she starts her story, I would like to give a very brief introduction on MMS technology and highlight some features and capabilities of MMS analyzer, AQS²pro. MMS is a protein characterization method that analyzes the secondary structure of proteins. As shown in the schematic diagram on this slide, there are three major components in the analyzer: a mid-IR tunable quantum cascade laser, a thermal electrically cooled detector, and a Y-shaped microfluidic transmission cell. The tunable laser generates optical signal almost 100 times brighter than the conventional light source in FTIR. The bright laser allows the use of a simple thermal electrically cooled detector without the need of liquid nitrogen cooling. The bright laser also allows accurate measurement of low concentration samples. Now we can measure samples at concentration as low as 0.1 mg/ml. While doing measurement, sample and the reference streams are injected alternatively into the flow cell, passing through the sampling spot. The absorbance of sample and the reference are measured almost simultaneously. Reference absorbance is subtracted from the sample absorbance in real time, and the reference-corrected absorbance spectra are collected. The real-time buffer subtraction and auto-referencing greatly enhance the sensitivity of the measurement and produce nearly drift-free signal. Compared to conventional FTIR, MMS improves about 30 times in sensitivity. Another very nice feature of MMS is the advanced analytical software package. This software is very user-friendly and provides straightforward automatic workflow and streamline data analysis. Based on MMS technology, Redshift-Bio, located at Burlington, Massachusetts, United States, has developed a novel protein characterization instrument, AQS²pro. So on this slide, I give a very quick summary on some features and capabilities of AQS³pro. So AQS³pro measures the amide one-band absorbance spectrum from 1600 to 1700 wave numbers and determines the secondary structure of proteins. It has 30 times improvements in sensitivity than conventional FTIR instrument. AQS²pro measures protein in solution with a wide concentration range from 0.1 mg per ml to over 200 mg per ml. There's no interference from excipients in buffer. And also in AQS²pro, one measurement, you can get 5 key analysis results, including the structure, similarity, stability, aggregation, and quantitation. Also, AQS²pro is fully automated with multi-sample capability and save up to 20 times in direct labor. And as we mentioned in previous slide, AQS²pro also offers streamlined data analysis process, which the users like it a lot. So let's get back to the story of amyloid peptide. So we have used AQS²pro system with a microfluidic transmission cell of 23.8 micron path length to test all these amyloid peptides that Dr. Daggett will talk in her story. We measure all these samples at room temperature. All the control peptide samples were measured at 0.8 to 1 milligram per ml in either PBS buffer or 5% DMSO in PBS solution, while 8 beta 24-hour and 120-hour incubation samples were measured at 0.38 mg per ml in 1:1 6 millimole sodium hydroxide to PBS buffer solution. The absorbance spectrum of amide 1 band from 1714 wave number to 1590 wave number were collected at a modulation rate of 1 Hz and a back pressure of 5 psi. All the data was analysis using RSB's data analysis software AQS data. Okay, so now I'm going to turn the webinar over to Dr. Daggett. So thank you for your attention, and please visit our website, redshiftbio.com, to find more information of MMS and its applications.
Hello, my name is Valerie Daggett, and I'm going to be telling you about studies that have been ongoing for many years in my lab at the University of Washington. And we've been studying amyloidogenesis, and amyloid diseases affect over 1 billion people worldwide. And they are fundamentally diseases in which a normal protein or peptide changes structure, aggregates to ultimately form amyloid, fibrils, and plaques. And so today I'll be talking about how we're trying to characterize the structures along the way. So in this cartoon depiction, you'll see on the far left the protein transthyretin, TTR, and it's a tetramer. It's non-toxic. It's a normal protein. It's a carrier protein primarily, and it's involved in heart failure when it aggregates, systemic amyloid disease. And so in the process of doing this, in going from its normal form to the fibrils that you see on the far right, which are non-toxic, it undergoes conformational changes. And it's been very difficult to impossible to characterize these changes in detail. So my lab started many, many years ago developing computational methods for mapping these conformational changes in atomic detail and to use those as testable models to work with experimentalists and to devise experiments to test what the computer simulations were showing us, because this is a very challenging problem, because you have the same entity changing structure and aggregating along the way. And so we really need a a microscope, a lens on that process to see in detail what's going on. And the first simulations in this area, we did in the early and mid 1990s, and that was on the A beta peptide associated with Alzheimer's disease, which I'll be saying a lot more about in a bit, and quite a bit with the prion protein and peptides. So we use this alternative approach to experiment to map this process. And in so doing, we started to fill in, as you see in the next slide, some of the gaps along the way. So from experiment, we couldn't see those conformational changes on the way. We could only see the starting and final states. So for transthyretin, it initially dissociates, and then there's a conformational change to lead us to the toxic infectious species, what are called soluble oligomers. Now, while the focus has been in the area on the fibrils and plaques, the amyloid itself at the mature end stage, it's really these intermediate species, these toxic soluble oligomers that are doing the damage 10 to 20 years before you see the plaques. And so this is where we were focusing our efforts. Now, in the next slide, you'll see what we find with molecular dynamics simulations if we take this same protein, transthyretin, and it's a beta sandwich. Imagine sort of two copies of what you see on the left there together, like two pieces of bread. And then just highlighting one here, we simulated it at low pH, which is what is used both in vitro and in vivo, triggers the amyloidogenic process. And as we did that, we, as you see in the next slide, the overlay is we saw a conformational change. And if you focus on that star, if you look at the two forms, it's flipped, so that when we look at on the left, say focus on the edge on the right of the left-hand structure, you'll see red, that's for carbonyl, jump down to a white, that's the NH group, the peptide bond, carbonyl. And you see this alternation of red and white. Whereas when you look over to the right-hand side, you'll see that all the carbonyl oxygens, the red atoms, are aligned. And then you go to the other side of the strand, and you'll see the white are aligned. And so this is a different structure, and it's caused by a peptide plane flip on every other amino acid. And it gives rise to a molecular dipole and interesting properties. So we called this initially gamma sheet. We switched to the name alpha sheet. It was discovered by Roger Arman in my lab, who you see there, and it was published nearly 16 years ago now. And it was a hypothesis. It was something that was just in the computer at that time. So we spent more effort on characterizing this structure. And in the next slide, you will see where this fits into the overall process. We hypothesize, we call this our alpha sheet hypothesis, that these toxic soluble oligomers contain this structure we discovered in the MD simulations. Now, I'll note that if we run those same simulations at neutral pH, we used low pH to trigger the process. At neutral pH, the structure remains intact. So when we then looked at more and more proteins, we found that all kinds of different amyloid proteins were forming this structure. So they were unrelated. We've got in the slide with a number of structures there, you'll see A beta, the amyloid beta peptide, which is involved in Alzheimer's disease. You'll see the prion protein, Creutzfeldt-Jakob disease, and systemic amyloid disease is transthyretin. You'll see superoxide dismutase, which is associated with Lou Gehrig's disease, ALS, and intrinsically disordered peptide, islet amyloid polypeptide, which is associated with type 2 diabetes. Now, what all these have in common is that they're non-toxic. However, just by looking at them, you'll see they're all different. They come in different sizes, different shapes, different structures. And so these are unrelated amyloid proteins with different sequences, different structures, different functions, different locations in the body. And yet, when we do simulations of these and others, we find that they transition into this structure that we initially found in transthyretin. They all form alpha sheet. So we hypothesized that then this is a common intermediate during the process of amyloidogenesis, which could then give us a handle on maybe stopping the process and neutralizing these toxic oligomers. Now, after the alpha sheet forms, it goes on to form the non-toxic fibrils. On the right-hand side, I won't be talking much about this, but it's now known that a number of bacteria make amyloid fibrils for stability, for what's called biofilms, to protect themselves from the host immune response and antibiotics. And we've also looked at the peptides and proteins from those systems: E. coli, Pseudomonas aeruginosa, strep mutans, Staph aureus, and others. And they also funnel through alpha sheet. And so we have a very general process going on here. And so if we distill that down in the next slide, you basically can come from many different structures: beta sheet, alpha helix, unstructured. You have different sequences, and you're funneling through alpha sheet and then going on to a common amyloid structure. Now, that gives us a common structural target for rational design. So now in the next slide, I wanted to spend a little more time on one particular system, the A-beta peptide associated with Alzheimer's disease. And you've likely heard that there have been a number of failures in the clinic with this. And we think it's because they're just targeting the wrong structure. So if we look at this graph, we're starting out with an unstructured peptide. It's small, 42 amino acids. And it goes on to form amyloid plaques. Now, the way we measure this in the lab is with a fluorescence assay, and that's what that curve is showing, is that you start out on some baseline, you're non-toxic, it begins to aggregate. We can see a change in fluorescence when the dye THT, thioflavin T, is bound by the beta sheet structure in the fibrils, and we see a rapid increase and then to another plateau after mature fibrils are made. So remember, this is a change in structure. So on the left, in the non-toxic form, we have something that's soluble. It's got biological activity, and it's involved in memory and learning and synaptic signaling. And it's changing in structure and then aggregating to become insoluble. And at that point, it has beta sheet structure. So while plaques remain the hallmark of amyloid diseases, they are not correlated with disease itself. So amyloid burden is not correlated with disease. There are patients that have substantial amyloid plaques, but they're cognitively normal. Instead, we know that there's something else going on before that, the toxic stage. And so if you're diagnosing or treating patients at the stage where the plaques have already formed, you're really too late. And so we really want to focus on these toxic oligomers. So these are small misfolded aggregates, oligomers. They are correlated with toxicity and disease symptoms. And so in the next slide, you'll see how this occurs in terms of the time frame. So we're starting out with something on the left that's normal and, as I said, involved in learning, memory, also neuroprotection. And then early in the process, in the so-called lag phase, where we've got the flat curve there, we see the toxic oligomers start to form, these structures intermediate along the process. They are soluble. They contain alpha sheet. We first proposed that many years ago. We've now demonstrated it, and I'll show you more about that in a minute. They form early in the process, long before you get plaques, but they also continue as the disease progresses. And then in this sort of one-way picture where we aren't continuing the disease, it will form early and then convert to amyloid plaque as you move up to the curve on the black. But remember, as the disease is really ongoing in a symptom or animal, there's continued production of the toxic oligomers. These oligomers are directly correlated with toxicity, cellular damage, clinical symptoms, and again, plaque is not. So in the next slide, I want to just go back to the details a bit, the structure. So from the simulations, we can carefully map out what the structure of alpha sheet is. And this was published some years ago. And what is shown is a Ramachandran plot, and it's telling us about the orientation, the structure of the main chain and peptide bond. And in conventional structure, you would have alpha helix, as you see in the lower left quadrant, beta sheet up in the upper left quadrant. And what's unique about alpha sheet is that it's alternating between two structural forms in this map. And so if you have beta structure, to make continuous structure, you'd have residue 1 in that region as defined by the arrow, residue 2, residue 3. You have repeating structure. Same with an alpha helix. Now, with alpha sheet, we alternate between local alpha R, alpha L, so right and left-handed alpha structure, alpha helix structure, but it does not form a helix. It does not wrap around because it doesn't have the repeating structure that you would need, where you'd need alpha R, alpha R, alpha R, for example, to form a slinky-like helix structure. So instead, our handedness is changing at each amino acid, and that's giving rise to this in effect, this peptide plane flip to give you the alignment of, as you can see, the red oxygens and white hydrogens in this sheet. So what we reasoned is that since we have a new structure, we want to test whether it actually exists outside of the computer, is we need model compounds. You can't deconvolute a spectrum or go and characterize structure without knowing how that fundamental structure behaves, its signatures, so to speak. And so we decided to make use of this alternating alpha L, alpha R structure to make model compounds small alpha sheet hairpins. And so the overlay shows the maps you get if you have L left-handed alanine versus left and right-handed alanine, L and D amino acids. And so when you have the alanine in the L form, the normal form, you see that it looks, it's populating primarily the alpha R region, whereas when you use the mirror image, the D form of the amino acid, you're populating up in the upper right-hand quadrant in the alpha L quadrant, which is relatively difficult for L amino acids to adopt stably. So we use this unique alteration and combine alternating L and D amino acids to increase the probability of forming stable alpha sheets. So again, to get these model compounds. So we designed hundreds of these in silico and ranked, ordered them, and chemically synthesized the best candidates. And in the next slide, you'll see one of our benchmark compounds, AP90 for our alternating peptide, AP90, and its counterpart, P90. So the difference between them, the sequences are shown. The difference between the two is only 6 amino acids. They have exactly the same sequences. However, 6 D amino acids are incorporated into the AP90, and you can see them by lowercase in red. So what's going on here is we have, as we start at the N terminus of the peptides, we have a strand, we have a turn in the middle where you see NEYSG, and then another strand. And so what you can see by the spectra is that they behave very differently. So first pass, AP90 is extremely soluble in water, in buffer, all kinds of solvents. We don't need organic solvents to solubilize it. However, P90, exactly the same sequence, but all L-amino acids, is very insoluble. We have to add organic solvent to work with it in solution. So on the left-hand counterpart there of the graphs, we've got circular dichroism spectra. And so we predict, because of the alternating right and left-handedness, the chirality, that we should get a null spectrum from AP90, which we essentially do. P90, however, forms a spectrum more characteristic of beta sheet structure with a bit of random coil. Now, on the right-hand side is FTIR, Fourier transform infrared spectra, in the film state for AP90 and P90. And what you can see there is that you get very different peaks. And the P90 gives a characteristic peak for beta sheet structure, whereas we have two different new bands for alpha sheet structure that give us a handle on deconvoluting spectra from amyloid species. Now, in the next slide, we had to show that really this structure was made by these peptides. And so we did NMR studies, 2D in solution NMR, and determine the structure. And we have many nuclear Overhauser effect cross peaks to do this. They give us ensembles of structures that are alpha sheet, and we do not get signals we would expect for alpha helix or beta sheet. And so if you look on the right-hand side of this, you can see the different components of this ensemble, and most of them are running right down the center, so you can get a view of this hairpin. So we call this one AP407. It will come back up in a second. Now I'm showing you how A beta aggregates over time. And so this is A beta 42. You can see the conditions for it. And we are monitoring in the black curve the binding of THT, the fibril formation. So you see a lag period, an increase, and then we get mature fibrils. And then the bar graphs are now showing toxicity. So we take those same samples at those different time points, and we're putting them on neuroblastoma cells and looking at cell viability as a surrogate for toxicity. And we find that this toxicity peaks in the lag period and then drops off as we convert to beta sheet structure and fibrils. So in the next slide, we just wanted to break down and see what the species were in this. So we have size exclusion chromatography shown on the right-hand side of the graph. And as we start with the black curve, we have monomer. At 8 hours, which you can compare over with the plot on the left, you can see the toxicity is increased. It increases further at 24 hours. And you can see that the curves, we get hexamer, what corresponds to hexamer, and then a buildup of something, a dodecamer. Now, then, if we go further out in time to 72 hours, you can see that we have a drop in the hexamer and a buildup in the dodecamer peak. Although I'll note that's the edge of what we can resolve with this particular column, and we're getting larger order, higher order aggregates there as well that are non-toxic. The toxic species are the red curves there, and they're primarily, as you can see, hexamer and dodecamer. So then in the next slide, we wanted to characterize this structure. So we went back to circular dichroism, and we expect, based on the alternating chirality, to have essentially null signals for alpha sheet structure. So we want to see if we get alpha sheet structure, going back to our 15-year-old hypothesis, if we're seeing alpha sheet structure in the lag period when the toxicity peaks. So if we start on the right and look at the black curve, you can see that the peptide is disordered under these conditions, and it's normal, it's intrinsically disordered peptide. As it begins to aggregate, you can look at that arrow going up from the black curve. This curve flattens out. It lifts and flattens. Now, it's not entirely flat because we've got more L-amino acids. We've got L-amino acids at the ends and in the turns. but it most definitely lifts and flattens out. From 8 hours, it lifts a little bit more to 24, and it becomes similar to that magenta curve, which is for the structure that we determine the NMR structure of, where we have high resolution structure. So now we go away from this alpha sheet flat region, and as it aggregates further, going to 48 hours in the intermediate where the sigmoidal phase of the THT curve lies, and then it starts to drop through 36, 48, we're forming beta structure, and then you can see beta structure in the blue curve corresponding to late in the process at 120 hours. So our toxic samples do not contain beta sheet, they contain alpha sheet, and the fibrils are beta sheet. And then in the intermediate, you get a mix. Now, you have problems quantifying null signals, however, going to the alpha sheet signal. So the next structure shows you, we started working with Redshift Bio. And so what's nice about this is we really want something in solution. So I showed you FTIR spectra earlier, but one thing we always worry about is in making those films to do the studies is that we're changing the conformation and that we're pushing it towards alternative non-toxic conformations. And so what's shown here is a number of spectra, the second derivative spectra for AP407, which is the one we have the NMR structure for, nice alpha sheet hairpin, an alpha helix model peptide, a beta hairpin model peptide in light blue and gray. And then two different A-beta samples from the same time courses you've been looking at. The red is the most toxic species, and the blue at 120 hours is from the plateau region. So we're definitely getting different spectra for the different secondary structures. And the fibrillar A-beta is a shifted beta sheet signal compared with the beta hairpin, which has been observed before. Now, if we take a closer look in the next slide and we subtract off, do a delta second derivative, we subtract off the AP407, and we've added different AP designs here. So these to give us a feel for the spread of what alpha sheet structure gives. So these have different sequences. And those are those bands in blue, black there. So now you can see as we subtract this off, we've got very distinct spectra for our alpha sheet designs versus the alpha helix model compound in the beta hairpin. So the MMS is able to distinguish alpha sheet from the other secondary structures. In the next slide, we add in our A-beta toxic form. And you can see it overlays very nicely with the alpha sheet designs and not the alpha helix, not the beta hairpin. In the next slide, we show now the sample from up on the plateau, and it is quite distinct. So this is, I think, very strong support. We've got toxic samples that contain alpha sheet. They track very well with our model compounds, and the fibrillar form contains beta sheet as we expect. So another thing we tried is we have this time course for the THT, and this is shown on the left of this slide, lower concentrations. So you can see the kinetics are a bit different if you're following that. But then what we did is we add our compounds, our alpha sheet designs, and the idea is since we know that we are forming the toxic oligomers in the lag phase, if we add our compounds, we should be able to bind to them through the complementary structure. And back to our fundamental alpha sheet hypothesis, we should be able to neutralize the toxicity. And so we neutralize this species and prevent further aggregation. So this shows that when we then add to A-beta, we add one of our compounds, AP5, you can see the curve doesn't rise. We nip the aggregation in the bud. What I'm not showing you here is we then have tested this in the neuroblastoma cells as well, and that's been published in that same PNAS paper cited there. And they do in fact neutralize toxicity. So these compounds, we've been able to show that alpha sheet structure is present in A beta, the A beta peptide as it aggregates, and that our compounds neutralize toxicity of that species through complementary binding to it and prevent further aggregation. The plot on the right shows you more of this, where we do the same THT experiments and then just quantify the amount of inhibition. So on the left, you can see it's 96% for AP5, and then you see now 3 other AP compounds with different sequences, and they also show good inhibition. These were also peptides that were used in the MMS study. Now, in contrast, we have 2 controls up there. So P1 is an unstructured random coil peptide, and P411 is a beta hairpin. They are showing no inhibition, so their curves look very much like they just overlay with the A beta THT curve on the left. So we are seeing statistically significant inhibition of aggregation with these peptides and not with the controls. So it is specific binding that's doing this. So in the next slide, just to wrap it up, we discovered a new protein structure in these molecular dynamics simulations. We proposed it's associated with toxicity, and I've shown you now through this talk some of our results supporting that, and there are others out there as well. The alpha sheet structure is characterized by a null signal by circular dichroism, so we see a lift up to this flat curve. However, it's problematic. We needed an orthogonal technique, something independent in solution to evaluate this structure. And although we had seen differences in film FTIR, we were concerned that we were altering the conformation in drying the films, and that method is more appropriate for fibrils. MMS then provides a nice alternative and can give us independent conformation of alpha sheet structure. And it has been really critical for these soluble oligomers because they're in solution. And so in the brain, in fact, they're diffusing around the brain. And so we really wanted a solution technique here and did not find others that were really giving us what we wanted. So next steps, we'll be looking, collaborating with Redshift to do, and Libo has already begun some of this, MMS of other amyloidogenic peptides and proteins, including the one involved in type 2 diabetes, Staph aureus, including MRSA infections, and transthyretin, which is what I began with systemic amyloidosis. So thank you very much for your time.
Thank you, Valerie, very much for your presentation. It's great to have you with us today. It's now time for our question and answer session. Both Libo and Valerie are with us to answer any questions that you may have. To ask a question, please type it in where it says type your questions here and then press submit. So, our first question. Are there any buffers typically found in protein formulations that are incompatible with microfluidic modulation spectroscopy?
I think I will take that question. So, so far, yes. So as we test different kind of proteins, peptides in different buffer, formulation buffer, and we did not encounter buffer excipients interfere with our signal. So all the common excipients has been used in the industry is we don't have any problem to measure samples in that buffer.
I see. Thank you very much for that.
You're welcome.
Our next question asks, do ApoE2 peptides have similar folding dynamics, and why is the E2 so protective?
Well, I guess I'll take that.
I'm not aware of anyone having looked at the ApoE2 peptides and their folding dynamics. And I think there are a lot of theories and ideas about how ApoE2 may be protective and modulate amyloid beta effects. But I'm not aware of any studies where people have really gone into detail and looked at the conformational properties of these. So that would be a good thing to follow up on.
Okay, thank you. Perhaps something for us to follow up on after the webinar. Our next question asks, is there any water vapor subtraction applied to the MMS spectra?
Yeah, I think I will take that question. So usually the big challenge when you use IR spectroscopy to determine the secondary structure of protein is the water band subtraction and the water vapor subtraction. And our MMS technology overcome that by using an auto-referencing mechanism, like I explained a little bit in my brief introduction for the MMS tool. The way we're doing that is we use a flow cell. So we measure the sample signal and the reference signal almost simultaneously. So we can do real-time subtraction of the buffer background, including the water or water vapor effects, so we can get a very clean subtraction of the buffer background. So we can get it subtracted very cleanly, and you can see from our data quality.
Thank you very much. Our next question asks, what characterization tools can be used to assess toxicity of oligomers and fibrils?
To assess the toxicity, well, the most common assay people use is one that we employ as well is to look at cell viability when you have toxic preparations applied to different cell types. And that's how we determine the toxicity in that plot, showing that the toxicity peaks during the lag phase before beta sheet is acquired. But there are a number of other assays one can use in these cell-based assays to look at that. There are also ways where you can look at, which I didn't show today, where you can look at how the oligomers affect cell signaling and monitor the toxicity in that way, because the oligomers have disrupt electrical signaling between neurons, and you can quantify that effect, and then you will see that our peptides are neuroprotective and will counteract the effect of the A-beta oligomers. So that's another way to do it. So there are a variety of different ways in sort of in vitro and in cell-based type assays to do that, aside from what you can do in animals.
Thank you. And following on from that, our next question asks how oligomers impair the cognitive functions of the brain.
All right. Well, that one is in a sense related to what I just said. And so one of the strongest correlates with disease, the oligomers and disease, is the way they affect synaptic signaling. And so one of the first steps is these oligomers attack the cells, and they affect signaling, and then they can also deform membranes and affect transmission of things across the membranes. And so there are a variety of different mechanisms at play, but one of the first steps is disruption of signaling.
Great, thank you very much for answering that question. Our next question asks, how easily is the upscaling of a possible assay for detecting small molecules to block A-sheet formation?
Upscaling. So I'm not sure what's meant by upscaling. So in terms of just maybe doing it in a high throughput manner and scaling up an assay in that way, you can make relatively stable alpha sheet oligomers of A-beta, for example, and that's described in our PNAS paper from 2019.
And from there, you can just screen different small molecules, peptides, whatever it may be. In general, small molecules have not, in our hands, been very effective because of the large surfaces involved to neutralize the toxicity. But it's fairly straightforward if you want to work in vitro with clean oligomers.
I see. Thank you. Our next question asks, how does the tau fit into or connect with this transient toxic oligomeric picture?
Yeah, that's an interesting question. So the A-beta effects are upstream of tau, and so we really want to hit it before the neuroinflammation, the tau phosphorylation, and the things, a number of deleterious physiological effects go into motion. And so that's why we've gone with A-beta. But also, tau forms its own toxic oligomers. And so we have proposed that they also contain alpha sheet, and we'll be looking at that. We'd like to knock it out before they ever form. Again, that's why we go after A-beta, but to the extent they're there, I think they probably do have alpha sheet. We've looked at 10 different systems now, and all of these amyloid systems we've investigated so far do go through alpha sheet toxic oligomers.
Thank you. Our next question asks whether dimers of A-beta 42 form A-sheet.
That's a good question, and I don't have the answer for that because in what we've done, we've never seen dimers. The smallest unit we've seen is a trimer in CSF from a patient. Otherwise, and it could be a concentration effect, I don't know, but we've never picked up dimers. If they, and their dimers are somewhat controversial. If they are toxic, then I would say they probably have some alpha sheet structure. But as I mentioned, the smallest we've observed under physiological conditions and is trimers, and that was in CSF.
Okay, thank you. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here and then press submit. Our next question asks, do you demo the system? I think that maybe is a question for Libo.
Yes. So we are demo system in the client set, and also we are testing clients' samples in-house. So like what we did for Dr. Daggett. So you can send us samples, we can test in-house, and if you're interested to run it hands-on our system, we can set up a demo, and then we can ship our system to your lab and then let you to hands-on our system.
Okay, thank you. Our next question asks, with the MD simulations where you first observed the formation of A-sheets, Valerie, did you only observe this when you used the disordered monomer as a starting structure, or did you observe any ability for beta sheets to transition back into alpha sheet structures?
Okay, so when we first observed this 15 years ago, a little over 15 years ago, we did not, it was in transthyretin, the tetrameric protein that was shown early on in the slides. And that process is triggered by low pH, both in vitro and in vivo, and in our MD simulation. And there's dissociation of the tetramer, and then conformational changes of this beta sheet protein. It's a beta sandwich protein, immunoglobulin-like fold, and it changes into alpha sheet. And so that is a beta sheet to alpha sheet transition, not a disordered to alpha sheet transition. And we've seen that many times in many different proteins now. Now, in root, before the alpha sheet kind of comes together and stabilizes, we do see flipping back and forth between beta and alpha sheet, and so it's quite a concerted process to get an entire sheet to go, and so it's quite dynamic. We'll shift back and forth until it kind of clicks in to alpha sheet. And yes, it is possible once it's in alpha sheet, to go back. Now, this is before it forms oligomers and when it's in the monomeric state and first adopting this structure. And we do see flickering back and forth in the MD simulations and in experiments. Thank you.
Thank you. Our next question asks, are there peptide characteristics of alpha sheets forming oligomers, including peptide length and intrinsic disorder regions? And can you predict it by bioinformatics?
Yeah, that's a great question. So we've looked quite hard at this, and one of the big factors in determining whether a sequence can form alpha sheet is the probability of adopting that alpha L local structure, which is, as I mentioned in the talk, difficult for L amino acids. So, and then as far as signatures for sequences, that's a hard one. And so there are some residues that are preferred relative to others, but they do not have to have lengths of intrinsic disorder. That's the case with transthyretin. It's a very ordered protein, and it goes from beta to alpha sheet. It's not intrinsically disordered and doesn't have segments of disorder. And so it's been very difficult to predict it from sequence alone. We've developed a number of, I have a bioinformatics lab. We've looked at this. We have the largest collection of dynamics simulations of proteins in the world, and we have mined those data, and we've developed libraries for protein and peptide design for rotamers and main chains, and looking at conformational propensities to address that question. And so far, I'd say there's no one single answer to that it really is, some residues are better at forming alpha sheet than others, but it's really in the context of the whole sequence.
Thank you. Our next question asks, has AP5 been tested in mice models for AD?
Yes, it has been tested in mice models. It's been tested in two different models. One is organotypic brain slices of a transgenic mice. That's in collaboration with Dr. Mike Mullen at the Roskamp Institute. And then also we did intracranial injection of AP5 into the live mice. And the peptides do indeed, under both of these situations, decrease the concentration of toxic oligomers. So in the sort of live ex vivo brain slices as well as in vivo, we do see an in vivo effect.
Okay, thank you very much. Our next question asks if THT can be a good assay for alpha sheet assessment, since THT binds to beta sheets with low affinity for other conformations.
So does THT, from all of our experiments, we don't see THT binding, or no, we don't. If it's there, it's very mild. There's very little of it. We don't see THT binding to alpha sheet. The THT binding comes in at the beta sheet when the beta sheet is formed, and so there may be some low affinity binding, but we have no evidence for that, and we have looked quite hard. So we see the alpha sheet come in and form before the beta sheet, and we can see the trade-off between them. So this is, as the alpha sheet comes in the lag period, we also pick it up in the CD, and we pick it up in the MMS under the same conditions as the THT assay is performed. And there is no binding of the THT to the alpha sheet species.
I see. Thank you very much. Our next question asks, how is the dock-lock process in alpha sheet?
I'm not sure what a dock-lock process is.
Okay, well, that's probably a question that we can answer offline if you prefer.
Okay.
I can move on to our next question since we have a number now coming through. Can you comment on the specificities of the monoclonal antibodies used in the clinical trials? And are there current monoclonal antibody therapies which target the toxic oligomers?
So the antibodies in trials now, or the trials that have ended recently in the last couple of years, have all targeted the monomeric state or the amyloid plaques or the protofibrils. And protofibrils contain beta structure. I'm not aware of any that can specifically target the toxic oligomers. And so they may get some non-specific binding here and there, but none of them can specifically target the toxic oligomers. There is, of course, you're probably aware of the A11 antibody from Dr. Charlie Glabe's lab. That does bind toxic oligomers, but it's also polyclonal, and it's not been, as you mentioned, in the clinical trials. It's not been in those clinical trials. So we are unique in being able to very specifically target the toxic oligomers, and we've got very tight sub-picomolar binding and sub-attomolar detection. So we're rivaling or getting tighter binding than antibodies and very specific for alpha sheet structure.
Thank you very much. Our next question asks if in vivo, what signals the conformation change in amyloid beta from a beta sheet to alpha sheet conformation? Do the amino acids change from L form to D form?
So in vivo, what signals the conformational change from beta sheet to alpha sheet? So in the case of A beta, we're going from disordered to alpha sheet to beta sheet. And with A beta 42, for example, the conformations are changing up to what a D amino acid would prefer, but the chirality per se is not changing. It's those L amino acids are not turning into D amino acids. But there are examples of D amino acids, very interesting examples within A beta where D amino acids have been inserted, and we think those are making it easier for the peptide to form alpha sheet and maybe contributing to the disease.
Thank you very much. Our next question asks, is neuroinflammation involved in the formation of A sheets?
So alpha sheets occur first, and they do appear to trigger. So in A beta, let's put it that way, in A beta, when it forms toxic oligomers, it does induce neuroinflammation, and that argues for alpha sheets having a role in that effect. Our designed peptide alpha sheets do not appear to induce neuroinflammation. So it's when you have the naturally occurring amyloid, alpha sheet amyloid toxic oligomers. Our peptides, in fact, neutralize that effect.
Thank you so much, Valerie. Well, I'm afraid that is all that we have time for today. Once again, I'd like to thank Valerie and Libo for their presentations and for answering your questions. I would also like to thank the webcast sponsor, Redshift Bio, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope that you'll join us again soon.